Vibrio, one of the gram-negative rods. Now, this type of organism is acquired by an external source, meaning that is present in the environment that can affect your gastrointestinal tract. Now, this genus consists upon three species. Now, the first one is Vibrio cholera, which causes cholera, Vibrio parahemolyticus, which causes diarrhea, and Vibrio vulnificus, which cause cellulitis and sepsis. Now, two of them have one source, meaning that they originate from one area. Now, both of these are acquired from marine life. So basically, what I'm trying to tell you is that this organism is hydrophilic, meaning both of these organisms require NaCl in order to grow. And similarly, they are acquired from such environments, meaning that Vibrio parahemolyticus is acquired from seafood and it results in diarrhea, which is actually self-limiting and goes up to 3 days. Whereas cellulitis and sepsis in Biochemical is when your fish handler or someone that is dealing in the fish in such or any type of sailor, when they have an abrasion on the skin, so from that abrasion, this organism actually gets inward and it leaves hemorrhagic bullae on that respective skin where the lesion has occurred. But treatment is quite easy with giving the patient doxycycline. Now we'll discuss our most prime bacteria, which is Vibrio cholera, which causes cholera. Now, this is a comma-shaped bacteria like all of the Vibrios, but it is divided into two categories based upon its cell wall O antigen. It's divided into O1 and non-O1. Now, O1 give rise to epidemic diseases, meaning it accumulates a large amount of population when it is occurring, and all of them have diarrhea. Now, non-A1 consists upon sporadic diseases, meaning that one or two patients can be susceptible and develop such a disease. Now, this O1 type of group has two biotypes, L-Tor and classic. This is further divided into many serotypes based upon its enterogenicity, meaning Ogwa, Inaba, and Hikojima. Now, how do Vibrio cholera get into your body? First of all, it is being ingested, meaning that it has an orophycal route, meaning that one organism is infected by a mouth and it can infect other organism because this organism is excreted into the feces. Now, the reservoir of this is shellfish, oyster, and shrimp. Still, marine life is acquired, but this organism is not halophilic, meaning that it can survive without NaCl. So when it enters the stomach, your stomach, basically acidic fluid kills it. But this organism will not die if you are taking proton pump inhibitors that inhibit the gastric secretion, or you have underwent gastrectomy in which half of your stomach is being cut and attached to the rest of this intestine, attached to the intestine. So there will not be a lot of acid secreting cells. So gastrectomy and PPIs provide an environment in which this organism can easily pass from the stomach to the intestine without being killed. Now, what happens in the intestine? It secretes certain type of enzymes which is called mucinase. Now, you know that there's a polypeptide glycoprotein layer on the intestinal cells which is made up of mucin. Now, this is a protective layer due to which a lot of bacteria over here cannot damage the endothelial cells. Now, by producing mucinase, there will be a break or there will be a breach in this glycoprotein layer. So now it comes to the infectivity part. Not all of the Vibrio choleras are virulent. Some of them which are actually virulent are because of a bacteriophage. Now, bacteriophage known as CTX. Now, this consists upon a certain DNA which gets encoded into the Vibrio's DNA. Now, when it is encoded into the Vibrio's DNA, it gives rise to a certain toxin to be produced by this organism itself, which is known as the Cholera Toxin. Now, this process of encoding bacteriophage DNA into the DNA of the organism or the bacteria is known as lysogenication. Now, when bacteria is able to produce toxin, this toxin actually consists upon two subunits, A subunit and B subunit. A subunit is with which the toxin is able to bind with the surrounding endothelial cells, and B is the B is actually the binding and A is actually the active unit. Now, what does the active unit does? It actually stimulates or bind to the growth G protein, which is GS protein, which is stimulating protein. Now, what it does is it by secondary pathways increase the cyclic AMP in the body. When the cyclic AMP increase, there is a lot of fluid which is present inside of the cell goes out of the cell, resulting in high volume diarrhea because a lot of cyclic AMP secreted, and this would affect the cells which are present beside it, and a lot of toxin would be secreted, and similarly this would result in massive watery diarrhea. The characteristic clinical features include that the patient would have rice water stools. The volume of the diarrhea would be quite large without RBCs and WBCs. There would be no abdominal pain, but due to large volume loss of fluid from the body, there would be dehydration. This dehydration is actually the dangerous thing which can lead to death by creating cardiac or renal failure in the blood, creating acidosis and hypokalemia, which is created by excess of potassium being excreted out from the urine. Now, what sort of labs do we do for it? In endemic cases where huge population is involved, we use our clinical judgment. In sporadic cases, what we do is we can do a culture stool and grow this organism as it is gram-negative on McConkey agar. But less colonizers are seen because this is a slow lactose fermenter. Apart from that, Vibrio is oxidase positive. It is oxidase positive. Another test, a biochemical test which we can do is that we can do grow it on TSI agar. What we see is an acidic slant and an acidic part, and hydrogen sulfide gas is being released. This is due to fermentation of sucrose by this organism. Another thing for rapid diagnosis is that we can agglutinate by polyvalent O1 or non-O1 antiserum, which we can detect in blood. So basically it is an agglutination test in order to identify the antibodies against polyvalent O1 or non-O1 bacteria. Now, treatment is that it normally is self-limiting. What we have to do is we continuously need to hydrate the patient because the diarrhea would result in dehydration, and that is the actual thing that is leading to death. So we need to replace water and electrolytes, but in order to replace it, we have to infuse the patient with glucose because it enhances the uptake of water as well as in electrolytes. Now, the antibiotic treatment for choice of this organism is tetracyclines.